Hello students, Mrs. Kynes here with your next section of notes over sex-linked disorders. These are going to be an application of things we have already learned. So let's first recap sex-determining chromosomes. We talked about this a bit when we discussed karyotyping and how to look for sex determination at the chromosomal level. If an individual has two X chromosomes, they are classified as female, which means that the females are homozygous for the X chromosome. When females produce eggs during meiosis, there's only one version of the sex chromosome that they can give to their gametes. So females only contribute the X chromosome to their egg cells. Every egg should receive just one X chromosome from mom. In male sperm cells, because males have the genotype XY, they get one X chromosome that always comes from their mother, and the Y chromosome, in the case of a male, the Y chromosome would always come from a sperm cell. But whenever the male cells undergo meiosis, 50% of the sperm cells will get an X chromosome from their genotype, and 50% of the sperm cells will get the Y chromosome. You see this worked out in a Punnett square on the right side of the slide. So for sex determination in reproduction, because there's a 50% chance the sperm cell carries either an X or a Y chromosome, that means there's a 50% chance the offspring will be female and a 50% chance the offspring will be male. So that is an important thing to remember regarding probability of sex-determining chromosomes and where they come from in meiosis. Most sex-linked disorders are inherited on the X chromosome. If you remember, also from karyotypes, the X chromosome was significantly larger than the Y chromosome. This is because it codes for more genes than the Y chromosome. The Y chromosome largely codes for male development or male reproduction. Most X-linked disorders or X chromosome-linked disorders are recessive, which means that for the individual to express the recessive allele, they'll have to have only the recessive alleles in their genotype. Most X-linked recessive disorders are rare for females because they get two copies of the X chromosome. So there's a chance that the female could carry the recessive allele, but it would be masked or covered up by the dominant allele, kind of like you see here. This female has the trait, but it's masked by the dominant healthy allele. Remember, recessive traits are where we can see the carriers. They can carry the trait and pass it to their offspring, but they themselves are not affected by that trait or disorder. So females may carry the diseased allele or the recessive allele, but she has the normal expression. The only way a female can express a sex-linked disorder or an X-linked disorder is if she is homozygous recessive for that gene on her X chromosomes, meaning that she carries both copies of that recessive allele. Now, it's a little bit different for males because males only get one copy of the X chromosome. So, from their mother, they can either get the healthy allele or the recessive affected allele. So, examples of sex-linked disorders you might want to commit to memory, because these are the ones that get used frequently, are colorblindness, muscular dystrophy, and hemophilia. Muscular dystrophy is whenever the muscles start to break down and get weak over time. This usually happens kind of early in life. Hemophilia is the inability to form blood clots, so the blood doesn't clot effectively after an injury. You can see 3 possible genotypes for females, but only 2 for males. So males have it or they don't. Females can have the disease or disorder, carry it, or be homozygous for the healthy allele. This is an example of a sample color blind test. And just to see, right, don't try to self-diagnose. Don't use this as an actual clinical diagnostic tool, but to test for color blindness, they give these individuals this type of figure to see if they can see the numbers in the middle. So if an individual cannot see the number, it is possible that for this test, they would have a red-green color blindness. On the left, you should see a 5, and on the right, you should see the number 2. These are other examples of colorblind tests. So with normal color vision, in A, you would see the number 29. In B, you would see 45. In C, you don't really see a number, just a bunch of swirls. And then D, you'd see the number 26. And if an individual had different forms of colorblindness, you may not see the same numbers. Okay, so the way you would set up a Punnett square for a sex-linked disorder is kind of similar to how we did with blood typing. You're going to notate your genotype base letters with the sex-determining genotype, and then the allele for being attached only to the X chromosome. So it tells you in this problem that Kelly is a healthy carrier of hemophilia. Okay, so it already gives you her genotype as a healthy carrier. Remember, this means she doesn't have hemophilia, but she carries a copy of the X chromosome that does code for hemophilia. So she's a healthy carrier. Adam has no history of hemophilia in his family. So this would be his genotype. Because he's male, he has an X and a Y. This is only found on the X chromosome, which is why there is no letter attached to that Y chromosome. So he has a dominant allele on his X. The question is going to ask you after you solve the Punnett square, what is the probability of a daughter with hemophilia, a carrier child, a child with hemophilia, or two healthy children? So let's go ahead and look at that. I've already been given the parent genotypes, so I can jump right into setting up my Punnett square. I have put Kelly's genotype on top of the Punnett square with two X's, and I have put Adam's on the left side with an X and a Y chromosome. When I fill out the Punnett square, I follow the same procedure I have with all other Punnett squares, where I pull the allele from the top and an allele from the left into each box. So one from the top, one from the left into every box. You can see here that there are both dominant X alleles in the upper left box. One from dad and one from mom. That means this individual will be homozygous dominant for the healthy trait. In the upper right box, this individual, I'm going to pull one from the left and one from the top, and this individual would be a healthy female, noting two X chromosomes, one with the dominant healthy trait, but this individual would also be a carrier. She carries the recessive allele on her second X chromosome. In the lower left box, there's an X and a Y chromosome. So this would be male, and they carry the healthy allele. In the lower right box, I've got an X and a Y again. So this would be male because of that Y chromosome, but he has the recessive allele on his X chromosome. So looking at my questions, it asked me what is the probability of having a daughter with hemophilia. So first I need to identify where the daughters are on the Punnett square. The daughters are right here in the top two boxes. Looking at their genotypes, it would be a 0% chance because neither daughter has only recessive alleles. To have a hemophilia daughter, they would need to have the genotype X lowercase lowercase h, meaning she is homozygous with that recessive allele on her X chromosomes. It asks you next, what's the probability of having a child that's a carrier? Well, the only child that can carry a recessive allele on their X chromosome is going to be a female, because males either have the trait or they don't, since they only have one X chromosome. So the chance of having a carrier child is going to be 25%, because only one out of the four boxes can produce a carrier. Next, what's the probability of having a child with hemophilia? You hopefully have already noticed that the bottom right box is the only one that has only the recessive allele. So that individual would have hemophilia. That means there's a 25% chance. And the last question, the probability of having two healthy children. So this one is a little bit of statistics and probability. So with each pregnancy, there's going to be a 75% chance that the mother would have a healthy child. So every time the couple has a child, we repeat that 75% chance or 3/4 chance that the child will be healthy. To solve this, it's a little different. It's not going to just be a 75% chance that they have 2 healthy children. We have to consider each pregnancy. So in each pregnancy, there's 3/4 of a chance or a 75% chance that the offspring will be healthy. So when you do statistical analysis on this, for each pregnancy, you've got to multiply those together to get the probability overall. So that would mean I take 3/4 times 3/4 for my 2 pregnancies, and there would be a 9 and 16th chance that they would have 2 healthy children. If you want to turn that into a percentage, it would be a 56% chance that they would have 2 healthy pregnancies by turning the percents into decimals, so 0.75 times 0.75, and then multiplying them together and timesing by 100 to put it in that percent form. Okay, that is it for video notes over sex-linked disorder.